is acted upon by the acetylcholinesterase, then it is converted into acetate and choline. Now, acetylcholine is actually stored in the synaptic cleft of the neuron. Say, at the base of the neuron, say if this is the cell, naka-whiteboard pen ka. Say, if this is the axon, this is now the tail part of the neuron or the nerve cell. Now, the acetylcholine is temporarily stored in the vesicles, which are found in the terminal part of the axon, tail part. But of course, that is produced by the cell body of the neuron. And later on, it will be stored in the synaptic cleft. Now, when there is impulse propagation, or when there is transmission of information from one neuron to another, the acetylcholine has to be released. And of course, that will transmit information to the next neuron. After transmission of information to the next neuron, it has to be degraded. Dili siya pwede nga magstay siya to the receptor molecule of the other neuron. Okay, say for example, okay, so you have here the vesicles, which stores temporarily the acetylcholine here. It's one of the neurotransmitters, of which, when there is, say these are the dendrites, now stimulus, now that will be received by these finger-like projections, and there is what we call as impulse propagation, until it reaches the terminal part of the axon, and then later on, these neurotransmitters, example, the acetylcholine will be released in the synapse and is now received in the receptor molecules. So, i-receive na po na siya din, and then, of course, that causes again signal to propagate here, and so on and so forth. Now, that neurotransmitter must not stay long in the binding of this receptor, or else the information here, so kanisya is always activated. So di siya magsigla na siya extend of that stage. So dapat mag-degrade ang neurotransmitter by this acetylcholinesterase. So dili siya pwede nga mo keep binding in its receptor. Or else, that is what happened, no? Magsige na lang, mag-emit og signal or action. The action is always there. So muna, if say, during muscle contraction, magsige kurog, or ana-ana mo hang diri, or say, it's in the muscle, so magsige lang contract imong muscle, dili na mo relax. That is what happened when the acetylcholine will keep binding on the receptor molecule. Sige siya elicit og signal. So, it has to be degraded by the enzyme acetylcholinesterase. So, pag degrade ana, balik na po siya. Recycle. No? Okay, so that's one example. We have lyase. Now, lyase, example, we have cis-aconitate. Now, cis-aconitate is one of the intermediate products in the Krebs cycle or citric acid cycle. Okay? So, when it is acted upon by aconitase, then that will be converted into isocitrate. Now, say for example, when pyruvate, say, after glycolysis, when pyruvate enters the Krebs cycle, then it has glucose 6-phosphate. So as what I mentioned a while ago, that you just say if this is the glucose, pag-ana mo, fructose na. By this enzyme, phosphoglucose isomerase. So you have glucose, alpha D glucose 6-phosphate. So that will give you another molecule, alpha D fructose 6-phosphate. Okay, phosphohexose isomerase. Then another, you have ligase. Ligase meaning joining of two molecules together. Okay, so the joining mo ni with this, you have the ATP, the L-tyrosine plus the tRNA. tRNA. So this is during translation process. So you need this enzyme tyrosine tRNA synthetase. Synthetase is the enzyme responsible for the synthesis. So you have the word synthetase. For synthesis, when you say synthetase, synthesis means formation. So you have here that becomes L-tyrosyl, pDNA plus you have AMP plus inorganic phosphate. So one phosphate is removed. Now we have to take note of these terms in the enzyme. So the whole enzyme is called the protein part of the enzyme is called the Apoenzyme. The whole enzyme, sometimes we call it the holoenzyme. Holoenzyme, meaning the whole part of the enzyme. Now this has different parts. So the protein portion of the enzyme is called Apoenzyme. Now though enzyme is protein by nature, but not all of the parts of the enzyme is protein. The protein part of the enzyme is called the Apoenzyme. Okay, you take note of this. The protein part of the enzyme is called the Apoenzyme. Now, the non-protein portion of the enzyme, which is metallic, non-protein and metallic, we call that as cofactor. But if the non-protein is called organic, so it is classified as the organic molecule, then we call that as the coenzyme. Coenzyme. Okay? Are you with me?
Yeah.
So you have the holoenzyme, the whole portion of the enzyme that acts as the catalyst. Now, the protein portion of the enzyme is called the apoenzyme. The non-protein portion of the enzyme, which is metallic, is called cofactor. Like for example, zinc, manganese. So that is cofactor. We term that as cofactor. So it interacts, or that is a constituent of the holoenzyme. So part na siya sa enzyme. Okay? Now, the non-protein portion of the enzyme, which is organic, like for example, B vitamin or vitamin B, mga vitamin B1, B6, they are called coenzyme because vitamins are organic molecules. So they are the non-protein parts of the enzyme, which is part of the holoenzyme for the whole portion of the enzyme. Are you with me?
Yes.
Okay. Now, the compound whose reaction an enzyme catalyzes is called the substrate. So, ang partner sa enzyme is the substrate. Mo interact with. Siya, ang substrate mo ay mo bind sa active site sa enzyme. Ang enzyme naa siya'y active site. So I'll show to you later, no, the picture of this enzyme. So the enzyme has its active site where the substrate binds. Okay? So that that is where the substrate binds. So it's the active site or the binding site. They're just the same. So that's the portion of the enzyme of which the substrate binds during chemical reaction. Okay, we have some example here. So this is just a schematic diagram of the enzyme. Enzyme substrate interaction. Okay, so this is our enzyme. The whole thing is the hollow enzyme. So you have here the hollow enzyme, or say maybe this one, except for you have the substrate, and then now this one is the protein portion of the enzyme, the apoenzyme. Okay. Now this one is the binding site or the active site of the enzyme. This is where the substrate binds. Okay. This is where the substrate binds. So you have here the substrate, and then this is the coenzyme. So coenzyme, if it is organic, like for example B complex. Cofactor if metallic. No? Metallic. Now you could imagine class kung walay coenzyme or walay cofactor. Kung bind ang substrate sa binding site sa enzyme sa may tabo, would it fit? No, so mo na kinahanglan taog, it could be cofactor or coenzyme. That's why they are essentials in our diet.
Pwede ba ilungan mo? Ilungan.
Dungan ang, depende yun sa need. Specific man siya. No? So, it is very, very specific to a particular. Say kung what is needed in a particular chemical reaction, kung metallic, kung metallic, say zinc ang kinahanglan. Say, for example, the growth hormone requires zinc, no? To elicit growth. So, ang zinc dito serves as the cofactor. Okay? Okay. So, you have here, kung substrate lang mo bind, luag, it cannot fit with the active site of the enzyme. So there's possibility that there will be no chemical reaction. If there's no chemical reaction, then there is no product formation. Are you with me? Okay. So you have also these terms, activation. So this is the process that initiates or increases the activity of the enzyme. So enzymes can be activated. So meaning initiating or increasing its activity. So there are some molecules that activates the enzyme, and there are some also which inhibits the enzyme. When we say inhibit or inhibition, this is a process that makes an active enzyme less active or inactive. So kung ang enzyme inactive na or less active, it cannot produce or it cannot say stimulates the product formation. It cannot do its job of speeding up the reaction. Are you with me? So dapat activated ang enzyme. However, dili pud pwede nga puro lang activated ang enzyme. There should be regulation. That's why nato na mga inhibitors. Say if there are too much products already produced out of the activity of the enzyme and the substrate, then there is a need of regulation. So, oops, daghan na kayo mo. So that is a message to the brain, or that's too much. Okay, so we elicit na po ang inhibitors. So kinahanglan tag inhibitor. That inhibits or slows down the activity of the enzyme. So there will be no more product formation. Kaya nga naman, nanubra na ang products. That is how our body is being regulated. Hindi pwede produce lang. No? Dapat slow down po. After products have been produced too much. No? So, okay. Now, we also have this word, okay, competitive inhibitor. Now, inhibitors can be competitive or non-competitive. So you have the word competition. You have the word competition, compete. So this refers to when a molecule or a substance is considered as competitive inhibitor. Now, it binds to the active site of enzyme, which prevent the binding of the substrate to the active site of the enzyme. Iyahang tabunan niya ang binding site sa enzyme for the substrate. So, dili na makabind ang substrate. That is competitive. Are you with me? Okay? Now, for the not competitive, this refers to any substance that binds to a portion